So up to Stephanophile area we have studied in the previous class. So today the today we will study the first family, that is Trichostrongyllidae. In that the genus are, first one is Trichostrongylus, second one is Ostratagia, third one is Cupria, and fourth one is Nematode, fifth one is Himoncus, and sixth is the Mystoceras. Mystoceras. So the general characteristic of this family are that in this family the worms are slender and devoid of leaf crown. They mainly parasitizes the alimentary canal. They are the parasites of grazing animals. So the first genus is Trichostrongylus. Trichostrongylus are also known as a black scoreworm. Their color is reddish brown. They have reddish brown color. In this, there is one structure, that is gubernaculum. Gubernaculum is present. Their eggs are oval and have a thin shell. In Trichostrongylus, the first species is Trichostrongylus colubriformis. Trichostrongylus colubriformis. It is present in small intestine of cattle, buffalo, sheep, goat. Another species is Trichostrongylus vitrinus. It is present in duodenum of cattle, buffalo, and sheep, goat. Another one is Trichostrongylus longispicularis. It is also present in small intestine of cattle, buffalo, and sheep, goat. Another, the last, the most important species in Trichostrongylus, because these all species are present in small intestine of cattle, buffalo, and sheep, goat. One species is Trichostrongylus tenuis. It is present in sika of fowl, turkey, guinea fowl, duck, and other birds. Second genus is Ostratagia. It is also known as a brown stomach worm. There is one specific lesion for the Trichostrongylus, that is, it causes ringworm-like lesions. Causes ringworm-like lesions. It also causes osteoporosis. This you have to remember. Another one is Ostratagia. It is also known as a brown stomach worm. It mainly occurs in abomasum of ruminants. It mainly occurs in abomasum of ruminants. Here the gubernaculum is absent, which was present in Trichostrongylus. Here gubernaculum is absent. In Estrategia, the species are Estrategia, Estrategia. It is present in cattle, goat, sheep, and horses. In abomasum. Another species is Eastertagia circumcincta. It is present in sheep and goat. Third one is Eastertagia pinnata. It is present in sheep. I am discussing only the characters of the parasites here. Their pathogenesis diagnosis I will discuss later when this family general characters are completed, when the specific genre characters are completed. So third one is Copria. It is present in small intestine of ruminants and also present in abomasum, but rarely it is present in abomasum. The species are Cupria curtisii, Cupria punctata, Cupria pectinata, Cupria oncophora. Fourth genera is Nematoderus. Nematoderus has circular mouth and the eggs of Nematoderus that pass out in feces are in eight cell stage eggs passes out in feces. This is very important point. Eight cell stage eggs passes out in feces. So in Nematoderus, on the shape of spicule, there are various species on basis of spicule. First one is Nematoderus spathi, spathiger. It is present in a small intestine of ruminants. Its spicule is spoon-shaped. Another one is Nematoderus bactus. It is also present in a small intestine of ruminants. Its spicule is heart-shaped. Third one is Nematodus follicolis. It is also present in a small intestine of ruminants. Its spicule is lancet shaped. Another genera is Himoncus. So Himoncus are voracious blood suckers. Himoncus are voracious blood suckers. Voracious blood suckers means that they requires large amount of blood, want large amount of blood as a feed. They occur in abomasum of grazing animals, mainly ruminants. Abomasum of grazing animals. Worms are reddish white in color. Here, spicules and gubernaculum both are absent. This one is very important. Spicules and gubernaculum both are absent in case of Himoncus. Here, genital opening is covered by vulvar flaps. Genital opening is covered by vulvar flaps in females. Species are Hemionchus contortus. It is present in abomasum of sheep, cattle, goat, buffalo. It is also known as a large stomach worm or wire worm or twisted worm or barber's pole worm. Here, the most characteristic feature which you have to remember in Hemionchus cotontus is that the ovaries are coiled around blood-filled intestine. Ovaries are coiled around the blood-filled intestine. Here, a male bursa has a characteristic asymmetrical dorsal lobe. Male bursa has a characteristic these 2 points are very very important in case of Hemionchus. Male bursa has characteristic asymmetrical dorsal loop with Y-shaped dorsal ray. So in OT, they may ask like that ki Y-shaped dorsal ray is present in which parasite? Then it is Hemionchus. Another species of Hemionchus is Hemionchus bubalis. It is present in abomasum of buffalo. Hemionchus placei, Hemionchus similis. These all are the species of Hemionchus. Another genus is Mysistocerus. Mysistocerus. The characteristic feature of this worm is that ki worm is thin anteriorly. Worm is thin anteriorly. Here cervical papillae are spine-like. Here also gubernaculum is absent. In that the species is Mysistocerus digitatus. It occurs in abomasum of ruminants. Then I will discuss about the general life cycle of the family Trichostrongyloid. So these all species, these all genus, all the six genus. So life cycle of Trichostrongyloid are is they are monogenous parasite. Monogenous, that means they have life cycle direct life cycle. So adult worm is present in abomasum or present in small intestine. Then eggs are released in feces. Generally, the eggs are unembryonated. They may be of 8 to 32 celled stage. Then, under favorable condition and environment, and there is a favorable condition. environment favorable condition like under optimum temperature, oxygen and humidity. The hatching of eggs occur. Hatching of eggs occur in environment and L1 is released. Then L1 feeds on soil bacteria. The bacteria present in the soil, the L1 larva will feed on the bacteria present in the soil. Then from L1, L2 is formed. Then from L2, L3 is formed, which is the infective stage. This L3 is infective stage in case of most of the nematodes. So L3 is the infective stage. Then this L3 will climb at the top of the vegetations. Suppose this is the vegetation, then this L3 will climb at top of vegetation. Then if then some definitive host consumes it during grazing, then it reaches the gastrointestinal tract. There they convert into L4 from L3, then L5, then adult. So this is the life cycle of trichostrongyloid. And here two things are important. Here the L4 stage, it is very important. The L4 stage in some species like Osterategia shows hypobiosis. Hypobiosis means arrested larval development stage. So L4 stage shows the hypobiosis, and when the spring season comes, in spring season, these L4 complete their development and become L5, then become adult, and the eggs are increased in feces. So this phenomena is known as spring rise, which I have already told you in that general parasitology part. So this phenomena is known as spring rise. And you have to remember that ki hypobiosis is also shown by hypobiosis is shown by first one is trichostrongyle, second one is Haemonchus, third one is Estrategia, fourth one is Couperia, fifth one is Nematoderus, and sixth one is Dictyocalus. So this is a very important point that hypobiosis is shown by which parasite? Then these all parasites shows the hypobiosis. So the pathogenesis of trichostrongylid infections, pathogenesis. So the trichostrongylid infections are generally known as hemocausis or tertagiosis, tertagiosis, and collectively known as GIT nematodosis. So in hemocausis, as I have already told you that it is a voracious bloodsucker, so it sucks ml blood per parasite per day. Per parasite per day. In this hemocausis, in hemocausis disease there are three forms. First one is hyperacute, second one is acute, and third one is chronic. Hyperacute form is rarely found. In this, there is hemorrhage and abomasum, severe anemia, and dark black feces. In acute form, there is decrease in PCV value. PCV value with PCV further decreases, then there is also an iron sorry, in chronic form there is an iron deficiency, edema. Due to the edema there is bottle jaw condition. So you have to remember that edema and bottle jaw condition is occurring in case of chronic infection, in chronic form of disease. And PCV decrease in case of acute infection, and there is dark black features and severe anemia and hemorrhage in abomasum and abomasitis also in case of hyperacute disease. There is one clinical signs in case of sheep, that is peeling of wool. Peeling of wool in case of Haemonchus infection in sheep. Another one is Ostatagia. In Ostatagia, you have to remember that there is one term which is asked about 2 or 3 times in where in last to last year, and continuous to 2 to 3 times in exam. This term was, that is Morocco leather. Morocco leather appearance and abomasum. So in Ostatagia, the two type of disease are found. Two types of disease are caused by the Ostatagia. First one is type 1 and second one is type 2. So the type 1 is mainly caused due to the adult worms. Due to the adult worms. And type 1 disease is mainly caused by the adult worms, and type 2 disease is caused by adult plus hypobiotic larva. So in type 1 disease, suppose when L3 larvae are available in large number in pastures and develop in adult worm, when this L3 larva is consumed by that host, then in the host there is development of adult. When developed in adult worm, that L3 larva develops in adult worm, the first type of disease is produced. This occurs in a very short period of time. Very short period of time, in around 18 to 20 days. And the second type of disease occurs in around 4 to 6 months. तो जब L3 स्टेज लार्वा पाश्चर में प्रेजेंट है, ग्रास ग्रास में प्रेजेंट है, उसको कंज्यूम किया है कैटल ने, तो सीप ने, कैटल ने कंज्यूम किया है, सीप ने मेनली, तो वो अडल्ट में कन्वर्ट हो जाएगा 18 से 20 दिन में ही, और टाइप 1 डिसीज प्रोड्यूस करेगा. और अगर इतने दिनों में नहीं कन्वर्ट हो पा रहा, देन इट प्रोड्यूसेस टाइप 2 डिसीज. तो टाइप 1 डिसीज इज मेनली caused due to the adult worm. This you have to remember. And type 2 disease is mainly caused by the adult and hypobiotic larva. Another thing is diagnosis. So for GIT nematodes, diagnosis, you can go for fecal examination. Fecal examination, you can see the eggs in the feces. But the most appropriate method for the diagnosis of GIT nematodosis is quantitative fecal examination. Quantitative, because nematodes are always present inside the body of the host, but they causes disease only when their numbers are increased. So instead of just confirming that this host has some parasite or not, it is much more important to confirm that what amount of load he is carrying, what amount of load that host is carrying. So in case of GIT nematodes, quantitative fecal examination is the best method to diagnose for the GIT nematodes. So in this, first one is egg per gram of feces. So it depicts the intensity of infection. Intensity of infection. Another one is increased plasma gastrin and pepsinogen also suggest the GIT nematodosis. Increased plasma gastrin and increased pepsinogen. This may be indicative of gastric nematodosis. There is one another important technique, that is Famacha technique. It is used in Haemonchus contortus. Famacha technique is used in case of Haemonchus contortus. इसमें होता क्या है कि इसमें पहले से it has a colored guided chart. इसमें कलर्ड गाइडेड चार्ट होता है. सपोज जैसे कि कोई शीप है, इसमें हम देखते हैं शीप की म्यूकस मेम्ब्रेन को, म्यूकस मेम्ब्रेन ऑफ आई. तो इसमें सपोज स्कोरिंग रहती है 1 2 3 4 5 ऐसे करके. तो सपोज 1 है और म्यूकस मेम्ब्रेन सपोज रेड है. यह है शीप की म्यूकस मेम्ब्रेन. तो सपोज म्यूकस मेम्ब्रेन है रेड और उसमें एक चार्ट रहता है, वो पहले से ही बना रहता है. तो रेड म्यूकस मेम्ब्रेन का मतलब है कि उसका स्कोर जो होगा वो 1 होगा. एंड दैट मीन्स कि उसमें हिमोंकस नहीं है. क्योंकि हिमोंकस होगा तो एनीमिया होगा, तो एनीमिया होगा तो जब ब्लड ही कम है तो म्यूकस मेम्ब्रेन रेड रह ही नहीं सकती. तो अगर म्यूकस मेम्ब्रेन रेड है शीप की तो उसमें हिमोंकस नहीं होगा, तो देयर इज नो नीड टू गिव एनी ट्रीटमेंट. और अगर सपोज 2 3 2 ऐसे रेड से कम होता जाएगा, सपोज इन केस ऑफ 5, तो अगर यहाँ पे व्हाइट है म्यूकस मेम्ब्रेन शीप की, अगर हम देख रहे हैं व्हाइट म्यूकस मेम्ब्रेन ऑफ शीप, तो जब इससे चेक करेंगे तो ये यहाँ पे व्हाइट कलर होगा तो इससे मैच करेगा, तो इसका मतलब है कि उसमें हिमोंकस है. इट इज very specific test for Haemonchus. It is a very specific technique for detection of Haemonchus in case of sheep. So you have to remember its name, that is Famacha technique. तो अगर यहाँ पे 5 है और व्हाइट म्यूकस मेम्ब्रेन है, सपोज व्हाइट म्यूकस मेम्ब्रेन है तो उसका स्कोर 5 होगा और यहाँ पे हिमोंकस है, और तो इन दिस केस वी गिव ट्रीटमेंट फॉर हिमोंकस. The Famacha technique is used for Haemonchus contortus, and in this we see the mucous membrane of eye of sheep and we match with the color guided chart. And for the treatment of trichostrongyloid, we use benzimidazole. Benzimidazole. And if the parasite is resistant for this drug, then we use monopontal. Monopontal for resistant nematodes. So this was all about trichostrongylide, and now we will discuss about lungworms. So in lungworms there are three genus. First one is Dictyocalus and Dictyocalus. Second one is Mularius. Third one is Protostrongylus. Fourth one is Metastrongylus. So these all are the lungworm: Dictyocalus, Mularius, Protostrongylus, and Metastrongylus. So first one is Dictyocalus. Dictyocalus is present in, अब lungworm है तो lung में ही present होगा, respiratory passage में होगा. It has a direct life cycle. Female are amphidelphic. Amphidelphic female. Species are Dictyocalus filaria. 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 It is present in bronchi and bronchioles of sheep, goat, and other ruminants. It is also known as a thread lungworm of sheep. Dictyocalus filaria is known as a thread lungworm of sheep. For it, the vaccine is Diffil vaccine. Diffil vaccine. And second species is Dictyocalus viviparus. It is present in cattle and buffalo. For its, the vaccine is Dictol vaccine. So for Dictyocalus filaria, the vaccine is Diffil, and for Dictyocalus viviparus, the vaccine is Dictol. These both vaccines are X-ray attenuated vaccines of Dictyocalus. Here, Dictyocalus viviparus cause one specific disease in cattle and buffalo. Last year, this question was asked in that ICR exam in match the column question. So, Dictyocalus viviparus causes the husk and hooves disease. Dictyocalus viviparus causes husk and hooves disease in case of cattle and buffalo. Here in Dictyocalus, you have to remember one thing, that the spicules are boot shaped. Boot shaped spicules. Third species is Dictyocalus anfieldi. It is present in equine. And fourth species is Dictyocalus cameli. It is present in camel. So the general, I will discuss the life cycle of lungworms. So I will discuss the life cycle of Dictyocalus and Mularius and Protostrongylus, and the last one Metastrongylus has somewhat similar life cycle as Dictyocalus. So in bronchi and bronchioles, embryonated eggs present. So the hatching occurs in bronchi, hatching of eggs in bronchi, then cuffed up. मतलब ये इंटेस्टाइन से आ रहा है, जैसे अपन ने वो पहले पढ़ा था ना, रेस्पिरेटरी रूट ऑफ माइग्रेशन, ट्रैकियल रूट ऑफ माइग्रेशन, वैसे ही कुछ है. तो हैचिंग ऑफ एग इन ब्रोंकाई होता है, देन Again, the animal swallows it. They are swallowed, and sometimes these eggs or larvae, which are present in bronchi, eggs or larvae, are also expelled in nasal discharge. Also expelled in nasal discharge or sputum or sputum or in sputum. So then it is swallowed. Then it reaches the intestine. Then L1 is also present in feces. This L1 has one characteristic structure that it has a small cuticular knob. L1 has a small cuticular knob. Then from L1, L2 will develop. Then from L2, L3 will develop. Then this L3 will reaches at pasture. Then if host grazes that grass, then it reaches to the intestine. And from L3, the L4 will develop in intestine, and L4 will goes to the lung. And in lungs, it become adult from L4. Then it reaches bronchi and bronchioles. Again reaches it again it reaches to bronchi and bronchioles. Then this is the life cycle of lungworm. So goats are more susceptible than sheep. Goats are more susceptible than sheep. Here, the clinical signs, there is one specific clinical sign, that is catarrhal parasitic bronchitis is caused by dictyocalis. Catarrhal parasitic bronchitis. Here there is one PM lesion, that is bunch of milk white worm with large amount of frothy mucus in bronchial tubes, also having desquamated epithelial cells. And for its diagnosis, the L1 larva has which I have already mentioned in that life cycle, that L1 larva has characteristic knobbed small knob is present in that L1 larva. So for diagnosis, in fresh feces, the L1 larva is seen under that microscope, and if it has a small knob, then it is dictyocalis. And for its control, I've already told you two vaccine dictyocalis. For dictyocalis, first one is Difil and second one is Dictol. Another one is Metastrongylis. Metastrongylis is a lungworm of pig and goat. Lungworm of pig. It causes parasitic bronchitis in pigs. Causes parasitic bronchitis in pigs. Species are Metastrongylis elongatus. Mainly the Strongylosis elongatus causes the parasitic bronchitis in case of pig. Just wait a minute. So, meta Strongylosis elongatus causes parasitic bronchitis in pigs. Other species are Metastrongylosis pudendotectus, pudendotectus, Metastrongylosis salmi. So the egg of Metastrongylosis has a corrugated surface. Eggs have corrugated surface. Life cycle is indirect. In the case of that Dictyocalus, the life cycle was direct. Here, the earthworm act as intermediators, and rest of the things are same as the life cycle of that Dictyocalus. Here only the difference is that ki earthworm act as an intermediate host, and the adult worm causes verminous pneumonia. Adult worm causes verminous pneumonia. This verminous pneumonia is caused due to the obstruction of bronchioles by adult worms. Obstruction of bronchioles by adult worms. It also causes consolidation of lungs. Another one is protostrongylus. Protostrong, Protostrongylidae. In that, there are two genera. First one is Mullerius, and second one is Protostrongylus. In that, the species is the general characteristic of this Protostrongylidae is that they occurs in bronchioles and parenchyma of lungs. तो lung worm है तो lung में रहेगा bronchioles में रहेगा. Then here another important thing is that gubernaculum is present. It also has indirect life cycle. So in Mullerius, there is one species that is Mullerius capillaris, which is also known as a hair lungworm of sheep. And there is one another worm which is also known as a hair lungworm, that is Venestrogylus, Venestrogylus pneumonicus, also known as a Biacolus pneumonicus. It is also known as the hair lungworm of sheep, but when you have to select among them which is known as a lungworm of sheep, then the most appropriate and correct answer is sorry, Mullerius capillaris. It is a lungworm of sheep. In Protostrongylus, the species are Protostrongylus rufescens, which is known as a red lungworm of sheep and goat. Red lungworm of sheep and goat. It has a boat-shaped. This one is important. It has a boat-shaped gubernaculum. Gubernaculum. Here, bursa is supported by chitinous plates. Bursa is supported by chitinous plates. Last one is Phylloids osleri. This is just for OT. It is not related to that order, but you have to remember that Phylloids osleri is known as a lung worm. It is a lung worm of dog. Here, one pathological sign is it causes hemorrhagic wart in tracheal bifurcation. Hemorrhagic wart in tracheal bifurcation. So this was all about the nematodes, and this was also all about the veterinary parasitology. From next class, we will study about veterinary anatomy or from veterinary pharmacology. I will tell you later in that WhatsApp group, or I will post in that notice board of this classroom what we are going to start next. So this was all about veterinary parasitology. If you have any doubt regarding that, what I have told you in whole parasitology, you can post in that notice board. And दूसरी चीज ये है कि पर्सनली मैसेज मतलब ऐसे रहता है ना, कई लोगों के मैसेज रहते हैं तो सबको जल्दी कुछ बता नहीं सकते क्योंकि चीजें मुझे हर चीज तो नहीं आती है, कहीं से ढूंढना पड़ेगा उसको या तो कहीं से देखना पड़ेगा। अब कोई अब क्वेश्चन भेजा है किसी ने, अब चार ऑप्शन है उसमें से चार में से कहीं ना कहीं से कुछ देखना पड़ेगा, फिर देख के बताना पड़ेगा, हर चीज तो आती नहीं और तुक्का तो लगाना नहीं है। तो हो सकता है कि जैसे किसी ने आज क्वेश्चन भेजा है उसका मैं रिप्लाई 3 दिन बाद दूं, 2 दिन बाद दूं, मतलब दूंगा जरूर, पर कोई ऐसा इंस्टेंट नहीं है कि किसी ने सपोज अभी भेजा है तो मैं उसका रिप्लाई आज ही दे दूंगा, क्योंकि वो मुझे भी कहीं से देखना पड़ता है और ये क्लिनिक्स जाना पड़ता है, टाइम नहीं रहता है वैसे इतना, पर मैं उसका रिप्लाई दूंगा तो 2 दिन 3 दिन बाद कभी भी मतलब वो मैं उसका रिप्लाई दूंगा। मतलब मैं ये कहना चाहता हूँ कि जैसे किसी ने अभी क्वेश्चन भेजा तो he or she should not expect from me that कि मैं उसका रिप्लाई 5 मिनट है 10 मिनट है फिर 1 घंटे 2 घंटे में होगा, दूंगा पर थोड़ा लेट भी हो सकता है। So that was all about veterinary parasitology, and what we will start next, I will inform you in that group. So I'm going to end this class.